Now this week, I'm going to tell you about how to write that DNA structures or gene structures by chemical characters. And also, I'm going to tell you about how to create or how to synthesize the DNAs chemically. So genes are the most important components of every living organism. We think we possess these genes in our bodies, but actually we were possessed by genes. Where does our gene come from? Of course, our parents pass on their genes to us, and we pass on our genes to our children. But humankind survives because many of us pass on our genetic information. But if you look at genetic inheritance closely, it is the genes that survive, not us. We are simply a mass of proteins. We become ash when we die. In some ways, the genes behave just like viruses. They ensure their own survival in our children even after we die. If I say it more precisely, our gene information will survive, not us. So we are simply vehicles to transmit the information. Because genes are made up of information, their structure must be designed so that the information they contain can be correctly transmitted or replicated. The information in our genes is written on a long double-stranded molecule we call DNA. The DNA information can be broken down into 4 parts. I'll show you the chemical structures. Adenine, guanine, thymine, cytosine. The chemical structures for the 4 bases are like this. They all consist of only 4 elements I told you about: C, H, O, N. And also they have ring structures that adds stability. I mentioned about it. Then the adenine and guanine are called purines. The thymine and cytosine are called pyrimidines. The more highly developed the life form, the greater the amount of information that is required in its design. Then, at the time that DNA bases were first discovered, the people saw that the complex life forms could not be expressed with only 4 different bases. However, all living things, from bacteria all the way up to human beings, have all 4 bases in common. Human beings should be better than bacteria. If bacteria have 4 bases, human beings may have 6. That's what people originally thought. But we have only 4, just like bacteria. As you know, the computers can do very complex jobs, but they use only binary codes, 0 and 1. Why? The computers use very long binary code information. The difference between human DNA and bacterial DNA is the length of information, length of DNA. To make a human being, it takes about 3 billion base pairs. Then there was one big mystery when these 4 bases were discovered. Dr. Chargaff first discovered at Columbia University in 1950, when a DNA strand is broken down, the numbers of A and T, or numbers of G and C, are almost the same at all times. In human DNA, for example, A is 30.9%, T is 29.4%. They are close to each other. Then G is 19.9%, C is 19.8%. Why does this happen? The answer lies in the structures of the 4 bases. That to solve this mystery, you must understand a chemical concept we call non-covalent bonding. In the next step, and in the next script, I will tell you about non-covalent bonding to solve this mystery. Stay tuned.